I think we can get started now. Looks like.
Okay, hope everybody can hear me. I'm in a little cave here and hopefully my internet connection will be good for a while. Today, we're going to have the doctoral defense of Paul Snyder. And Paul will be my 34th student who hopefully will receive his doctorate today. Paul has a really diverse background, probably more diverse than any of my students. He hails from Florida. Out of high school, he didn't go straight to college like many of us did. Instead, he went and got a job at Toys R Us and Babies R Us, where he was for 9 years. And he became the manager and he made actual money. And while he was dealing with Toys and Babies R Us, he was in a metal band. He's a heavy metal guitarist called Primitive Astronauts. He's got a lot of skills. Babies R Us actually gave him a scholarship to get his associate's degree, which he got. And then after about 9-10 years, he goes back and gets a bachelor's degree at the University of South Florida. And he worked with Jason Rohr and Lynn Martin, who some of you may know. After his bachelor's degree, he then became a research assistant at the Carey Institute for Ecological Studies with Rick Osfeld, who some of you might know also. That's where my former student Barbara Hahn is also. And then he became a lab manager for Vanessa and Zenwa at the University of Georgia for about 2.5 years. And he said he loved that job. Okay, now he then decides he wants to go back and get a doctorate. And I guess I didn't answer some of his emails, so he got really upset with me. And finally, we connected and we're together. So he gets his graduate work done, supposedly right here, and he has a job lined up. He's going back to the University of Georgia with Vanessa, where he's going to be an ideas manager. And I asked him what that meant. And he said something like he's going to do statistical coding for all the students in ecology or biology, and started to explain that. And I said, you didn't have to explain that because I taught him everything he knows about coding. So we're good there. So he's got lots of experience. He's also a superb video gamer since grade school. And some of his friends say that they actually have contracted with Sony for a video game. I don't know if that's true or not. Finally, or almost finally, he's a gourmet food aficionado. If anyone ever eats with Paul, he only likes the best food. His typical breakfast is McDonald's Egg McMuffins. His lunch is Taco Bell's or Jersey Mike's. And his dinner is frozen pizza. And he's still in a band. His band is called the Brutal Bridges. I don't know if you can see that. There he is. He plays a mean guitar with Clint Epps, Carmen Harjo's best friend, Timmy, and some other people. But most importantly, he's a really good guy, really nice guy, and he's a really good scientist. He's driven to do really good work. And today he's going to talk about a study that shows diversity and disease interrelationships. I know we're all sick of hearing about diseases, but this is really good stuff. And it's one of the rare experimental studies on diversity and disease. So that's what I'd like to say, and I'll let Paul take over from now. Go ahead, Paul.
Awesome. Thank you for that introduction, Andy. Appreciate it. All right, let me make this window on my screen a little smaller so I can see my slides. Okay. All right, well, thank you everyone for being here today. I'm going to walk you through some of the work I've done during my PhD here. But before we get started, I'm going to give you a bit of background information. Let's see if this thing will go forward. There we go. All right, so as many of us are probably aware, we're currently experiencing global biodiversity declines with accelerated losses recorded across taxa. These declines have been called a biodiversity crisis, or the sixth mass extinction event, with wildlife population extinctions by some estimates over 1,000 times pre-human background extinction rates. And changing diversity can have numerous effects on an ecosystem. We can all imagine adding or removing a species could change numerous things: food webs, species interactions, predator-prey relationships. but we're going to be focusing on how diversity changes alter disease dynamics. So one of our key concepts, let's get rid of that. Here's going to be that changes in diversity can alter disease transmission. And
Paul,
so,
I have to interrupt you. You have to share your screen.
oh no. Sorry, everybody. Embarrassing. All right. Is that better? Can we all see it now? Oh, there's chats. All right. Okay, we got the technical difficulties out of the way, hopefully. So we missed a few slides. My apologies. We were here. One of our key concepts is going to be that changes in diversity can alter disease transmission. So how do disease and diversity interact? There's been a few different relationships defined or suggested in the literature, one of which is the dilution effect. A dilution effect is as host diversity increases, disease risk decreases. The opposite effect has also been cited, amplification. As host diversity increases, disease risk increases. Trying to find a general rule for disease and diversity. These are both probably too simple, and it looks like the reality is more complex. These are three different shapes of diversity disease curves from recent literature. We don't really know if there is a defined shape that holds true for disease and diversity, and if it would be specific between different pathogens, hosts, or systems. So how do disease and diversity interact? We're going to focus on one metric, host competence, which is the ability of a host to transmit a given pathogen. So I have some examples here, and I made them frogs since I'm working with frogs. If we have a low competence host, it's able to be infected by a pathogen, but it may be very poor at transmitting this pathogen. A moderately competent host, after being infected with a pathogen, would be able to transmit the pathogen, and a highly competent host would be an effective transmitter of the pathogen. Hosts vary in competence, though, by species, as well as potentially by populations. And increased diversity increases the variation in host competence. So as we add species, we may get species that are very competent or that are incompetent to a certain pathogen in an ecosystem. These variations in host competence then can alter the disease dynamics of a system. So hopefully I sold you on the key concept that changes in diversity can alter transmission of diseases. And our next key concept is going to be that disease can also alter diversity. So there's some reciprocality here. As part of the biodiversity crisis, amphibian populations are declining worldwide, with nearly a third of amphibian species estimated to be at risk of extinction. There are lots of factors involved in these declines. I'm sure many of you in the audience are thinking habitat loss, and absolutely, you're right. But amphibian populations declines are especially influenced by diseases. Amphibians are hosts to at least 3 globally distributed emerging infectious diseases, including Batrachochytrium dendroptitis, Batrachochytrium salamandrivorans, and a group of viruses known as ranavirus. Amphibians are also hosts to trematode parasites, well, to lots of parasites, but the trematode parasites have been shown to be able to have population level effects. So we have a lot of important pathogens here involved in amphibian declines. And amphibians are an ideal model for examining these disease diversity relationships as amphibian diversity is threatened. Disease affects amphibian populations. We have decades of amphibian field data And we're able to experimentally manipulate different aspects of disease, both in the laboratory and field. So with that background out of the way, I'm going to introduce you to our 3 focal pathogens. These are the 3 different pathogens I worked with during my PhD, all of which potentially affect the amphibians in our system, and all of which have worldwide impacts. These 3 are Batrachochytrium dendrobatitis, which we can refer to simply as BD, ranavirus, and trematode parasites. I'll walk you first through BD. Batrachochytrium dendrobatitis is an aquatic, globally distributed fungal pathogen. It causes the disease chytridiomycosis, and it's been shown experimentally to be able to infect over 40% of amphibian species. On the right here, we can see the complex life cycle of Batrachochytrium dendrobatitis. I'll walk you through it starting here at this motile zoospore. This zoospore is flagellated and able to move towards its potential host. It does this by chemotaxis towards keratinized tissues. Amphibians, adult amphibians, post-metamorphic amphibians, have keratin in the skin, and that's going to be what these feed on. I work primarily with larval amphibians, and in our tadpoles or larvae, the keratinized tissue is in the mouth parts. So here it says burrows into the skin. It could also be mouth parts. So as zoospore moves towards our host, it burrows into the skin. It matures into a sporangium, which fills with zoospores, and then they're released, completing the cycle. BD interferes with electrolyte transport, which causes nervous issues and eventually leads to cardiac arrest. Our second globally distributed generalist pathogen we'll be looking at, ranavirus, is a true generalist able to infect amphibians, fish, and reptiles. Here we have a drawing on the right, and here we have an image of ranavirus inside of tissues. Ranavirus is able to invade most cell types, making it both very contagious, and it creates a systemic infection, which leads to hemorrhagic ulcers and death. The final pathogen group we'll be looking at, and I'm just calling our parasites pathogen for ease of diction, are going to be trematodes. 2 groups of trematodes, a Kinostoma species and the genus Ruboroiae, have both been shown to have population level effects. And for our purposes, they're shed from infected snails. I'm going to walk you through the complex life cycle of trematodes really quickly. Just get a sip. Okay. We start the cycle with our definitive host, which is infected with an adult worm. The definitive host is going to release eggs, a waterfowl, and its droppings into a body of water. The egg is going to develop into a myricidium, which is a motile form of the egg, which is then going to seek out its first intermediate host, a snail. The trematode ends up taking over the gonads of the snail, effectively castrating it, while producing the next generation's sporocyst and rediae. These sporocysts and rediae, which fill the snail, the primary purpose is to produce these cercariae, which are going to leave the snail then and infect our second intermediate host. The cercariae is motile. It moves towards the host, and once it comes in contact with the host, it sticks onto the skin, and then it kind of inches towards its target location. Maybe it more flops towards it. But the target location is dependent on species. For Ruboroia, shown here, the cercariae is going to move towards developing limb buds, where it insists, which will cause the type of multiple limb malformations shown here. The echinostoma species, on the other hand, will make their way to the cloaca and then go up into the urethra and encyst in the kidneys. When the cercaria gets to the target location, it's going to burrow into the tissue and drop its tail, forming this cyst we call a metacercaria. And now to complete its life cycle, this cyst needs to be eaten by the definitive host. Often, these infections have effects on the host, which reduce survival, potentially making it easier for our definitive host to eat them and complete the life cycle. So for our 3 pathogens of interest, what does the literature say about these disease diversity relationships? For BD, Trichochytrium dendrobatitis, the literature suggests a dilution effect. Both of these studies by Kat Searle and Barbara Hahn, were done with the same system that I'm working in, and these are laboratory experiments which suggest a dilution effect for BD, which would be as host diversity increases, disease risk decreases. For ranavirus, we don't have a lot of information in the literature about diversity. The information we do have is along the lines of infection by multiple strains of ranavirus or co-infection with ranavirus and another pathogen increases the risk of ranavirus, but we don't have any host diversity information to speak of. And then for trematodes, there's a little formatting issue. For trematodes, we also expect a dilution effect. This Johnson examination of California wetlands in Nature found a dilution effect with trematodes based on a non-random arrangement of host competence in wetlands. In addition to host diversity, though, we could also vary pathogen diversity, and co-infections are actually the norm in the wild. A recent paper here by Stutz et al., 2018, surveyed 90 wetlands and found that 80% of animals were host to at least 2 pathogen species. By animals, I mean specifically amphibians. Sorry. Co-infection can alter disease progression, severity, and transmission, and relationships between disease and host diversity may be altered with changes to pathogen diversity. So those are our pathogens. Now I'm going to introduce you to our host species. Our 3 host species are all amphibians of the Oregon Cascades mountain range. And Anaxyrus boreas, the western toad, is our focal host for all of the experiments that I'm going to show you here. Anaxyrus boreas is declining across its home range. It's been impacted by disease. There's some population extirpations in Colorado that have been attributed to disease. And they also have a long history with a variety of trematode parasites. Our other 2 amphibian hosts in the system are Rana cascadi, the Cascades frog, and Pseudocris regilla, the Pacific tree frog. So I'm going to show you a bit about how these different hosts may overlap. They occupy different altitudes, and A. boreas is going to be our highest altitude species, found only above approximately 2,400 meters. Rana cascadi then, that's found a little bit lower. And Pseudocris regilla has a very large range, occupying most of this. Hopefully, from this little approximated diagram I created, you can see that there's opportunities to have 1, 2, or 3 hosts at a given wetland, depending on the altitude and conditions of the same. So those are my pathogens and my hosts, and now we can start walking you through my 3 chapters. The Blaustein lab is an experimental lab, so I'm going to be showing you a series of experiments, starting with some experiments to try to answer the question, does increased host diversity reduce ranavirus disease risk? The first thing we need to know to answer this question is a little bit about our host competence. So what do we know? Not much for ranavirus, unfortunately. So we start with a laboratory examination. This laboratory examination was done in concert with the Rohr Lab at the University of South Florida and was led by Chloe Ramsey, who came out here to Oregon to work with us on these paired experiments. For the laboratory examination, hosts were raised from collected eggs. We do this so that we can be sure none of these hosts have previously been exposed to one of these pathogens, as that may change their response. And then each of these species, our 3 host species, were individually exposed, 28 of each, to 10 to the 2 plaque-forming units per mil for 48 hours. Plaque-forming units is a proxy measurement of concentration for viruses. The idea being that in 1 mil of this solution, if we spread it out on a plate, we could get 10 to the 2 plaques, viral plaques. And for this experiment then, we followed our infected or exposed hosts through metamorphosis and then 3 weeks post-metamorphosis. Here's an image of the experiment in progress. The experiment started with all of the animals in these cups. These are 1 liter cups to which they would be fed shrimp flakes ad libitum. Upon the emergence of front limb buds, the amphibians are moved into these plastic shoebox containers at a 30-degree angle, which allows us to have water at the bottom, but then also an area for the amphibian to climb out as it metamorphs. The animals once moved into these shoebox containers were fed vitamin-dusted crickets ad libitum. So what did we measure here that I'm going to share with you? Survival, days post-infection, and pathogen load. Let's see what we found. For the survival analysis, we'll see days post-infection on the x-axis, and we'll see the proportion surviving on the y-axis. First, for A. boreas, we'll see that there was no difference between our ranavirus exposed and control animals' survival. For Rana cascadi, we also found no differences in survival between control and ranavirus treatments. However, for P. regilla, we did find significant differences in survival, with the majority of regilla dying 10 days, within 10 days of exposure. We also then shipped the kidneys and livers to Purdue University, where the Hooverman group ran PCR to get viral loads of ranavirus infection, so we could tell how infected they were. And we learned that our A. boreas did have ranavirus present, but at levels too low to quantify. This suggested it likely tolerated the infection. Our viral load for cascadi was absent. We did not detect any ranavirus. So this may have either resisted the infection or cleared the infection before they died and were sent for processing. And finally, P. regilla did have quantifiable loads of infection. And so if we want to infer host competence from this, we could infer that P. regilla is our most competent host. After infection, it does have quantifiable loads of the virus present in the body. A. boreas then would likely be our next most competent host, as it did show some presence of ranavirus, and Rana cascadi would be the least competent host most likely, as it was not infected here or cleared the infection. So with this information now, we're going to examine disease and diversity in this large-scale mesocosm experiment. This image is the experimental setup, and each of these tanks holds 120 liters. To each of these, we added a bed of sterilized leaf litter, and then we added water seeded with algae and phytoplankton, and we allowed these to sit for 3 weeks before experimentation. So by the time we started, these were kind of like green, natural-esque ponds. And again, for this experiment, we have raised our hosts from eggs we collected in the field. Here we're using the same ranavirus exposure dose. It was only a 36-hour exposure here, though. It was 48 hours in the lab. And then the actual experimental treatments, we have 2 levels of host diversity, either 1 or 3 hosts present in a mesocosm. And then we have 2 treatments, either unexposed, control, or exposed animals. And the way we do this is we have a group of naive animals, or animals which have not yet been exposed to any pathogens, already inside of those mesocosms. At the experiment start, we add 3 animals which are either unexposed or exposed to the pathogen. So then these animals are going to be responsible for transmitting this pathogen through the mesocosm. With the addition of these 3, we'll have a total of 36 animals per mesocosm. And here we're just going to be looking at the measurement proportion surviving. We check this at 2 time points, 30 and 60 days. And let me show you our axes real quick before we talk about this. On our x-axis here, we have our exposure treatment, either control or ranavirus. Our diversity treatment will be denoted by either a solid or open circle. And on the y-axis here, we have the percent surviving of just our focal host, A. boreas. So after 30 days, we found that in our 3 host treatments that were exposed to ranavirus, we had reduced survival. So A. boreas survival was reduced in 3 host treatments exposed to ranavirus. If we consider all of the host species present, we see the same pattern. Now, the experiment ran for 60 days. By the experimental end at 60 days, we do now see some mortality in the 1 host ranavirus exposed treatments, but we have had a complete collapse of our populations in our 3 host ranavirus treatments. This is for A. boreas. Now, if we consider all of our species together, we see the same pattern. And in fact, after 60 days, in all of our replicates, we had a single animal surviving in the 3 host ranavirus exposed treatments. I do see a question and answer thing at the top. I think I'll get to that after. Okay. Sorry, I distracted myself with that little blip. So what did we find out from that? From our laboratory experiment, our conclusion is that only P. regilla appeared to be susceptible to ranavirus, and that P. regilla appears to be a competent host. In the mesocosm, all 3 hosts appear susceptible. Host diversity was associated with increased mortality, an amplification effect. And P. regilla, as a competent host, is likely responsible for the increased disease risk to the remaining species. Okay. For chapter 2, host diversity and BD, we asked the same question, but this time with batrachochytrium dendropatitis. Does increased host diversity reduce BD disease risk? And again, we want to know, do we know anything about host competence? Yes, we do. Many amphibian species have been experimentally exposed to BD individually. In our system, A. boreas is expected to be the most susceptible and has been documented to experience mortality from BD in as little as 48 hours. Laboratory examinations of our system by Hahn and Searle of the Blaustein lab suggest that BD disease risk will be reduced with increased host diversity. So I'm going to walk you through quickly some of Searle's results so we can see what that laboratory dilution effect data looks like. We're going to start with A and C. Similar to the graphs we just saw, A is just considering A. boreas, and C is considering all species. On our x-axis, we have a number of letters here: 2A, 3A, 6A, 3A, 3P, 3A, 3R. The A here is Anaxyrus boreas, our focal host. The R is Rana cascadi, and the P is Pseudocris regilla. So this is the same system that we're working in. The number in front of this is the number of animals. So these are replicates with 2 A. boreas. These are replicates with 3 A. boreas, 6, 3 A. boreas, 3 Pseudocris regilla, etc. So for A and C, we're looking at viral load. And I modified these to make this legible, so you're not seeing the units, genomic equivalents, etc. the viral load was measured in. When A. boreas is alone, viral load is higher than when A. boreas was combined with other species. This is true both considering just A. boreas or considering all species. Searle also looked at infection prevalence, the proportion of hosts that were infected, and we see a similar pattern where we have high infection prevalence. It's like over 80% when it's only A. boreas present, but as we add additional species, infection prevalence drops. So we've now conducted the same experiment, another mesocosm examination with identical parameters, except this time with BD instead of ranavirus. Again, hosts were collected, raised from collected eggs, and we used a BD exposure of 10 to the 3 zoospores per mil for 36 hours. Just like before, we have 2 diversity treatments, 1 or 3 hosts, and we have 2 exposure treatments, control or BD exposed animals, which are then added to an assemblage of naive animals. Again, we're measuring the proportion surviving. So what did we find? After 30 days, we found A. boreas survival was not different in any treatment. This was also true if we considered all species present. After 60 days, however, we had reduced survival in BD exposed 1 host treatments. This was also true when we considered all species, not just A. boreas. So our conclusions from our host diversity BD experiment: that host diversity reduced mortality in BD exposed treatments, a dilution effect. Mesocosm scale results here matched our laboratory examinations and supported previous research. And even though this host system was identical to the ones used for the ranavirus experiments, we found the opposite relationship between disease and diversity. Okay, chapter 3. This is our final chapter I'll be showing you. Does pathogen diversity alter the host diversity relationship? Here we decided to not only alter host diversity, but also pathogen diversity, this time including all 3 of our pathogens, to see how pathogen diversity affects these host diversity relationships. This is a laboratory examination, as pictured. Again, we raise hosts from eggs collected in the field so we can be sure they did not have previous exposures. This time we needed to acquire trematode parasites, which needed to be collected from ram's horn snails. Ram's horn snails. These were collected from Finley Wildlife Refuge. And once we returned with the snails, we put them in these 6-well plates, screen them under a dissecting scope for their parasites, and then we sort them and house them based on the parasites they're infected with. All of these snails are fed spinach ad libitum. And our most prevalent trematode species present were Echinostoma species, and so Echinostoma are what we used for these experiments. Here we have a little bit more complicated experimental design, but I'm going to walk you through it. Again, we have 2 levels of host diversity, either 1 or 3 hosts. Here, instead of just a control or treatment group, we have 8 different treatments. Control, each pathogen alone, and then every combination of pathogens. And then this time, I wanted to look at the early time periods that we kind of missed in our big 30-day mesocosm gap. And so this was run in triplicate with one set of treatments going for 2 days, another set of treatments running for 7 days, and a final set running for 14 days. So we could try to parse some effects across time. And for these are all 10-liter aquaria with 9 naive animals. And here we've only added 2 aboriuses, either BD exposed or not, or ranavirus exposed or not. The trematodes then were added directly to the tanks within an hour of collection. And these trematodes are viable to infect amphibians for about 6 hours after they're shed from the snails. We also used the same pathogen loads we used in previous experiments for these, 10 to the 2 plaque-forming units for ranavirus and 10 to the 3 zoospores per mil for BD. Here we measured a number of things that I'm going to share with you. Mean mortality, growth rate, which we've measured as change in SVL per day. SVL stands for snout vent length, and it's a standardized length measurement for amphibians and lizards. And then development. And we measure development with something we call Gosner developmental stages. And these are an amphibian-specific standardized set of developmental milestones. So what did we find? Well, we found a number of things. First, and I'm going to show you, just pause real quickly. I'm going to show you a lot of information here, but I'm only going to highlight a few things because there's just so many treatments to look at. Here on our x-axis, we're going to have our various treatments paired with our 1 and 3 host versions for comparison, with mean mortality of the 9 naive animals that started there on the y-axis. And what we found was that we had one treatment with significant mortality, and that was the BD and trematode treatment with 1 host diversity. So with only a single host present, we had a statistically increased mortality in the BD trematode treatment, but not in the 3 host BD trematode treatment. Some of these look like they may be different, but we don't actually have a ton of statistical power here. Okay, our next measurement is growth rate. Again, there's going to be a lot going on in these figures. On our x-axis, we have each of these treatments paired with our 3 durations side by side, either 2, 7, or 14 days. And on our y-axis, we have the change in growth per day in millimeters. The asterisks along the bottom are going to be treatments that were different from control. So here we can see growth rate in our trematode, BD and ranavirus, and BD, ranavirus, and trematode treatments with 1 host all had growth rates different from control. You also probably noticed that on our day 2, we have these really long error bars and some additional symbols. The square is denoting that day 2 is different from day 14, and the X that day 2 is different from day 7. I'm just going to highlight one of these quickly to show you how these can be different. Here, actually, I'm going to back up slightly, sorry. You may notice also that in day 2, they're all really big. That is not incredibly surprising, these error bars, because amphibians are very plastic. They can change growth rate, development, coloration, behavior, etc., to changing conditions. And all of these animals were just placed into new experimental conditions. So even our control group has to adjust to new housing and potentially new conspecifics. But the direction of this change does change by treatment. And so here we have the highest growth rate in our trematode exposed 1-host group. If we look at our 3-host trematode exposed treatment, though, that effect is gone. Likewise, if we look at our highest growth rate in our 3-host treatments, it's BD and trematode exposure. But in our 1-host treatments, that effect is not present. So we're seeing that not only is each different pathogen treatment altering outcomes, but the combination of host and pathogen treatments are altering outcomes. Finally, with development, we have our treatments here displayed simply on the x-axis and average change in Gossamer stage on the y-axis. Again, it's not really important we understand the Gossamer developmental system, just that it's standardized and we're measuring change. Okay, so we found in our 1-host treatment, 3 different treatments had delayed development: BD and ranavirus, trematode, and ranavirus and trematode treatments. All experienced delayed development. In our 3-host diversity treatments, though, only one group, BD and trematode, experienced delayed development. And this group did not experience delayed development in our 1-host systems. So what are our conclusions for chapter 3? Well, there are no overarching patterns here. It's very important that context matters. Exposure to both BD and trematodes increased mortality when A. boreas was the only species present, but not when A. boreas was housed with other species. Growth rate is most variable immediately after exposure to a new stimulus. The direction of changes in growth rate appears dependent on both host and pathogen diversity, and some combinations of host pathogen diversity cause delayed development. So overall, I'm going to take you back through our 3 chapters real quick to wrap up our conclusions. Chapter 1, we asked, does increased host diversity reduce ranavirus disease risk? And our answer was no, it did not. P. regilla amplified disease risk for other host species, and species that were not susceptible in the lab were susceptible in the mesocosm. In chapter 2, we asked, does increased host diversity reduce BD disease risk? It appears to, yes. Here we found in the same system as the ranavirus experiment, we got a dilution effect with increased diversity instead of an amplification effect that we saw previously. And here our results matched previous laboratory examinations. Finally, in chapter 3, we attempted to ask if pathogen diversity can alter the host diversity relationship. And I think we answered yes, but it appears that these relationships are very context dependent, with specific responses based on the specific host pathogen combinations. And that is my PhD research. Thank you. I have a lot of people that I need to thank and acknowledge. I think I'll just leave this up for a moment instead of reading through it. But I couldn't have done this without help from an endless number of people, everyone here, as well as many, many more people, everyone in the department, everyone who's been in my life, keeping me sane, etc. I really appreciate all of you. This is certainly something I wouldn't have accomplished alone. Okay, and now I'll take questions, and it looks like there's some already waiting for me. Seasonal variation and response in chapter 1. Oh, that's a great question. So in chapter 1, we did not. Chapter 1 was conducted entirely during the summer. And so it was conducted in a, oh, I've got 2 chat windows now. I'm sorry, guys. Okay, chapter 1 was completed during the summer. I'm sorry here. So we do not have any seasonal information. There would likely be differences by season. One of the most important abiotic factors in amphibian disease is often temperature. Temperature appears to limit BD, and it changes the rate of transmission for rhinovirus. First question, what are the causes of declines of Tarica? I'm not sure I know what Tarica is. I'm just going to Google it. Is this the first name of one of my species? Oh, newts. I didn't focus on salamanders or newts at all, but I would assume the declines are similar to those for our rough-skinned newts as our other amphibian species. We're going to have habitat loss through human land use changes, pollution to waterways, and of course, disease. Awesome. Okay, we have another question here. Was the mortality seen? Oh, and you guys can't see the questions, can you? I need to make sure to read these. Oh my goodness. I'm sorry, everyone. Was the mortality seen after 30 to 40 days in experiment 1 really mortality or just metamorphosis? If the former, why do almost all die? Oh, in the laboratory. It was metamorphosis. We had a large amount of death in our metamorphs. And so even the controls, we experienced death, whether it was they had trouble eating the smallest crickets. Well, they didn't though. It honestly seemed like about 2 weeks after metamorphosis, a lot of them would give up and just stop coming out of the water. We did change our protocols for future experiments that Roar Labs now used, where they add a moistened towel to the bottom, which the frogs seem to like more than the plastic base. Great questions. Do we have any other questions? Got another minute or two we could do this. Okay. Oh, was I supposed to click the answer live thing? This is my first seminar. I'm sorry.
That's okay, Paul. You don't have to do that.
All right. Well, I think that's it then, right? Oh, we got one more. Here it comes. What is the most common infection pattern in the wild? Mosaic. No, I don't know. I'm not sure what you mean by infection pattern, Emily. I'm sorry. Oh, do you mean like that relationship, dilution, amplification?
Yeah.
That's hard to say. The question, what's the most common infection pattern in the wild? If we're talking about disease diversity, there seems to be bias towards publishing dilution effect research. So it's hard to say from the literature what's really most common. But there is a lot of evidence for large-scale dilution effects. There's also some evidence that disturbances lead to dilution effects. And so in a lot of these situations where we're losing diversity, we may see something like that. Cool. Oh, there's more. I didn't scroll. Okay, from John Romancic, very interesting patterns, especially in chapter 3. Does one of the patterns in chapter 3 strike you as most interesting? And can you explain why? Ooh. Let's scroll back and look at them. I thought these were really interesting. The growth rates, this wasn't something I really expected to find anything super interesting in. I really hadn't analyzed it at first, assuming there wasn't going to be anything, and then there ended up being a whole bunch of actual differences. So this ended up being really interesting, and then the fact that we see so many different directional changes and differences between our 1 and 3 hosts, I thought were also pretty neat. Although it does mean if you wanted to follow up on this, there would be an awful lot to tackle, right? You'd have to maybe choose one of these pairs and then try to parse out what's making them different. Okay, great. Well, thank you again, everybody. I appreciate it. Oh, wait, we got what was your favorite part of your project and why? My favorite part of my project was probably training undergrads. So I had a big army of undergraduates. Each field season, I had at least 5 undergraduates working with me. We had thousands of animals to keep alive in the lab, not only for my work, but for all the other grad students' works. We needed to go collect those animals. And it turns out I like teaching, and I like getting students involved in science. So that ended up being the most entertaining for me. My favorite part was definitely training undergrads each year. Another great question.
Okay, I'm going to have Andy come back on and just wrap us up, and then we will head out.
Okay, Paul, good job. Now we'll do the rest of it. Thank you, everybody, for attending.
Okay.